I didn't. And in the signal detection theory, they have a threshold for this. So whenever the internal signal is higher than the threshold, you're going to say yes. And if it falls below, you're going to say no. So if you say yes and the stimulus was there, it's called a hit. And it can, of course, happen that the stimulus was there, but because of the stochasticity, it just fell short of the threshold. So you'll say no, it's called a miss. And if there is no stimulus, the internal representation of signal strength on average is going to be lower. And you're going to sample from a black distribution. And so typically, you'll say no if there's no stimulus. It's called the correct rejection, but occasionally because of the internal noise, it may happen that you have something that is above the threshold and you'll say yes, even though there was no stimulus, and that's called the false alarm. Okay, so I think most of you know this stuff. It's interesting to consider what happens if you present a stronger stimulus. So then the green distribution shifts to the right because the internal representation of signal strengths will be higher. And if you then copy the thresholds, you'll see that you have quite a number of false alarms here. So you might want to raise the threshold a little bit, decrease the number of false alarms. You will not get many misses because the signal is strong. But if you then copy this threshold to a situation with a weak stimulus, you see that now you get quite a number of misses. And this is what the signal detection theory describes. So it's sort of a trade-off between false alarms and misses. And that also depends on how bad it is to have a false alarm and how bad it is to have a miss. For instance, if you're working in, you know that very well in Israel, if you're working in an airport and you're scanning suitcases, to have a miss is really bad. You know, there will be somebody with a weapon. I'm not sure who it is. Switch off the audio because there are some noises. Can you see who it is? Anyway, so if you are working somewhere else and a miss is not so bad, then you might not be so concerned, right? And have more have less false alarms and more misses. So that is basically what is well described by the signal detection theory. And the question that we asked is in our study is where is then this internal signal that is compared against the threshold? What causes the variability, basically the width of these distributions? And what is this threshold? What is this threshold? What does it correspond to in the brain? And when we started to work on this, we soon realized that our work has impact on theories on consciousness. And one of them that seemed to fit quite well is this global neural workspace theory. So I'll say a few words about that. So here, if you present a weak stimulus, it's going to be propagated to higher areas. And then in this theory, there's sort of a mysterious process that is called ignition. And if ignition occurs, then there is a reverberation between higher and lower areas. And I'm now thinking of it as a memory trace. Okay, so if this ignition occurs, the stimulus can be weak, then there is a memory, say persistent firing for the stimulus that you just saw, even though the stimulus is no longer there. So if the stimulus goes out, the frontal cortex might still uphold a representation of the stimulus that you just saw. And now, according to this theory, there could be some variability in the feed-forward propagation of the activity. So sometimes a stimulus may not reach the frontal cortex in sufficient strength, and then it will not give rise to ignition. It will be forgotten. And sometimes the same stimulus can reach the frontal cortex with enough strength, and then it will give rise to ignition and a working memory. So that was the idea. Turned out that was a very good theory because this is basically what we found. So what we did is we trained monkeys on basically the same tasks you've been doing. So they look at the fixation point. We present the stimulus on 50% of the trials. And if they see the stimulus after a delay, they make an eye movement to where the stimulus had been presented. And on the other 50% of trials, we did not present the stimulus, and there was always this reject dot. So if the monkey wanted to report that he didn't see a stimulus, he could always make an eye movement to this reject dot. And this shows you the probability of seeing the stimulus as function of contrast, which is on the x-axis here. So if the contrast is high, of course, the monkey always sees it, about 100% correct. The animals were on the staircase where we gradually kind of reduce the contrast. At some point, they drop, of course, and we define two thresholds: the high threshold where the animals were 80% correct, and a low threshold when they were 40% correct. If it was below the threshold, it was a really difficult to see stimulus. Between the thresholds, it was intermediate, and above the high threshold, we said this is a really easy stimulus. Okay, and then we kind of grouped responses across different recording sessions based on these three categories. And we recorded from V1, V4, so that is sort of intermediate level visual area, and dorsolateral prefrontal cortex. Now, I should warn you that the neurons that we recorded from in dorsolateral prefrontal cortex are also involved in the generation of eye movements. So we are not going to dissociate the pure conscious percept from the plan to make an eye movement here. Okay, so that's a caveat that you have to keep in mind. Now, when we started to record from primary visual cortex, we were in for a bit of a surprise, because what we are showing here is the three categories. Of course, easy stimuli have higher contrast, so you get a larger response. Difficult stimuli have weak contrast, so you get a lower response. But here we're starting trials based on whether the animal saw the stimulus or not. And we matched the contrasts. So this difference between seen and missed trials is not based on the difference in contrast. Contrast was the same. But you see that on the missed trials, there was weaker activity than on the hit trials. As if on the pathway from the eye to the primary visual cortex, there is already a loss of information. And that was completely unexpected, at least in my mind. So apparently there's already quite some variability in the information processing going from the retina to primary visual cortex. Now, another thing that you may notice is that the easy missed trials, here in red, have a higher level of activity than the difficult trials that were seen. That's also important because it demonstrates that a certain level of activity in V1 is no guarantee that that stimulus is going to make it into conscious awareness. Okay, apparently information can get lost downstream from area V1 on its way to conscious awareness. Now then we did the same experiment in area V4 and we got basically the same results. So also in V4, the missed trials with easy contrast gave rise to more activity than the hit trials with the low contrast. So also here in V4, a certain level of activity is no guarantee that the information will reach conscious awareness. And in dorsolateral prefrontal cortex, we might be reading out the eye movement, of course. It's very different. So you see that here, it's really a sort of a bimodal distribution of activity. Whenever the animal sees the stimulus, there's a high level of activity. And if the animal misses the stimulus that was there, there is a higher level of activity, even if the contrast is relatively high. You see there's kind of a rise of activity, but somehow it does not reach, maybe there's a threshold somewhere, right? And if the activity doesn't reach that threshold, then the activity will subside. So we also looked at those trials in which there was no stimulus. And in black are the trials where the animal had the correct rejection. So he reported that the stimulus was not there. See, activity is always low, but something interesting happens on the false alarm trials. So here the animal makes an eye movement reporting a stimulus that was not there. And you see that this ramping of activity and activity eventually reaches the same level as the hit trials. So what we think is going on here, but we can't prove, is that there are sort of spontaneous ignitions, spontaneous ramps from a low activity level to a higher activity level. And our suspicion is that these spontaneous ignitions happen at different time points on different trials. So, and if you average many of these quick ramps, but they happen at different times, then in the average, you will see this slow ramping of activity. Okay, now we can't really demonstrate that, but I'm going to show you results from a model, and that is exactly what you see in the model. Okay, so if of these brain regions, there's one that would be compatible with the internal representation of signal strength, it's obviously dorsolateral prefrontal cortex. Now here we're not dissociating it from the eye movement, but an interesting question for future research is whether there are other areas, maybe also in posterior parts of the brain, where you have this similar sort of bifurcation behavior, maybe even response modality independent, right? So, but that's something that needs to be investigated. Okay, now I talked to Stan de Hane, I think 5 years ago or so, and he got really excited because he said, that's exactly what I always simulated and predicted. And you know what I'm going to do? I'm going to make the simplest of models to try to reproduce your result. And that's what he did. So he made a brain that consists of 5 neurons. 5 neurons in total. And these neurons are stochastic. There's one LGN neuron that propagates its activity to one V1 neuron that has a self connection and has a V4 connection to V4. And there are V4 connections to parietal and frontal cortex. And you see here, there's also feedback connections. Now he made the connections, so activity propagates, and he then makes the connections between parietal and frontal cortex extra strong, so that if these neurons reached a certain minimal level, they would be able to self-sustain their activity, giving rise to a sort of bifurcation. Okay, so that if that happens, we can take away the stimulus and the activity will stay in the network. So that is what you would call ignition, and that would correspond to a hit trial. Now, because of the stochasticity of the neurons, the same stimulus may sometimes stay below the threshold for the self-sustained activity, so there will be no ignition. And if you then take away the stimulus, activity collapses again, and that would be a miss, according to the model. So you can basically reproduce most of our results with this very simple model. So here, for instance, you see that on hit trials, there's more activity than on miss trials. Well, that's just stochasticity. And if there's just more activity in V1, the probability of a hit, of course, is higher than if the activity in V1 is lower. You see also that the activity on the easy miss trials is higher than the activity on the difficult hit trials, because on some of the trials, activity just gets lost downstream from area V1 in the model. Here in black are the correct rejections, and in blue are the false alarms. And in the model, we see these spontaneous ignitions. So what happens is, that happens only maybe on 1 or 2% of all the trials. Actually, that was also the case in the monkeys. And then occasionally it happens that just because of this strong recurrent activity between parietal and frontal cortex, and there are some spontaneous fluctuations, it can happen that just by chance, it goes across the threshold for self-sustained activity. Now this happens at different time points in different trials, and then you see a very quick ramp. But if you then average them across trials, you see the slow ramping. Okay, so in the model we can really see that. And it's quite interesting to compare the model to the neurophysiological data, and also a bit embarrassing because I think Stan made the model on a rainy Sunday afternoon. And for us in Amsterdam, it was 4 years of tough work. So this is the asymmetry between modelers and neuroscience and neurophysiologists. Okay, so some conclusions here. Basically, we see that the variations in internal signal strength are well explained by variation in V4 processing. That seems to be just a stochastic process. The threshold for perception is this bifurcation threshold that kind of pushes the model into a state of self-sustained activity. So that is the threshold for creating a working memory. And we saw that these false alarms can be explained by spontaneous ignitions. Okay. So now I would like to shift gears a bit, but if you have questions about this, maybe it's a good time to ask them.
Yeah, Pieter, I actually have a question I was curious about. I don't know if you can see me or not. So I really love this study, but I was always wondering about this in a sense, you know, here you had a very brief kind of perceptual stimuli, and yet we maintain awareness of things that are there despite the large, widespread reduction in the firing rates. Okay, so both in bold, well, and in the neural firing rates, we see when there's something you continue looking at, there's a reduction of the neural activity, and still we are aware of this. So how do you think we support this continual awareness despite reductions of the firing rates? Is this frontal area kind of maintaining a working memory representation, and all the back of the brain has already gone back to normal?
Yeah, I think we will see that in the next part of my talk, actually. So in some case, there is a continuous interaction between higher areas and lower areas, and that may indeed explain that situation. Yeah, I agree. Yeah. So maybe it's good to move on then to the next part, so which is on perceptual organization. So if you see this picture, you see a lot of stuff, and your visual system quickly organizes it into objects. So for instance, you don't have a hard time seeing that this leg and this nose is just part of the same zebra. And that's far from trivial, because vision starts with fragmentation. So the visual scene is analyzed by local processors, neurons with small receptive fields in early levels of the visual system. And somehow our visual system has to kind of glue back together the fragments into coherent object representations. And people used to think that this was done by synchrony. Well, that turned out to be completely wrong. So it turns out, and that's what I've tried to convince you of, that this is solved by the labeling of image elements with enhanced activity. And psychologists call this process object-based attention. Okay, so object-based attention is responsible for binding. And so what I will argue, we call this the incremental grouping theory, that if an image like this is presented, then the visual brain creates this image where all those image elements that belong to the object you're interested in are labeled with enhanced activity. So now we're going also to consider the feedback connections and lateral connections, because we think that they are responsible for the labeling of image elements with enhanced activity. So I'm going to test a little bit of this. So please pay close attention. I'm going to briefly present the stimulus and try to recognize what is there. Okay, who saw what was there?
I can guess. I only saw something furry at the middle of the picture. Maybe a squirrel.
Yeah. Did you see some colors too? What colors were there?
No, but I'm not representative.
Okay. Here it is again.
Oh, it's a bird.
It's a bird, yeah. But it really takes a bit of time before you recognize it, right? So I think this is a very informal illustration, but I think you first see that there's stuff, maybe colors, and then later your visual system organizes it into a representation of a bird, and that takes some time. Okay, so that's what I wanted to talk about, and there is a very simple version of such a figure ground task that has been studied by Victor Lamme first, before him by others, but in V1, I think first by Victor. And already in 95, so that's more than 25 years ago, he demonstrated that the response elicited by image elements in the receptive field of a V1 neuron that belong to a figure are enhanced relative to the same image elements. You see, he carefully had exactly the same image elements into situations in the receptive field. But if the receptive field fell on the background, activity was lower. And the idea is that you first get a feed-forward response, and then later you get this response modulation, and that this is caused by this recurrent processing, maybe even feedback from higher visual areas. That was the hypothesis. Okay, so I want to revisit this. So I want to discuss briefly how this works or how this can work in the brain. I would like to discuss the role of attention. I would like to demonstrate that this late V1 activity is really important for perception and that it depends on feedback connections because that had not been shown or clearly demonstrated. So first, a little bit of background on this process. So if you want to know how texture segregation works, it's a good idea to look at the classics. So here are a few of them. So people in the psychological literature typically propose that there are two processes, one that is sensitive to boundaries, so the orientation discontinuities that happen at boundaries, and another process that's sometimes called region filling or region growing, where if you select some image elements of a particular orientation, you may want to co-select adjacent image elements with the same orientation because, of course, they're likely to belong to the same perceptual object. Now, if you then want to make neural networks, people have looked at this, and it turns out that if you want to do boundary detection, it's a good idea to put inhibitory connections between neurons tuned to the same orientation. And so here's a few studies that use that strategy. So if you're a neuron in V1 and your receptive field is in the background, you're tuned to the right orientation, all your neighbors are active, and they're going to supply you with a lot of inhibition. They're going to suppress your response. Same will happen if you're tuned to the left orientation and you respond to the center of the figure, and only if you're close to the boundary, inhibition comes from only one side, and therefore inhibition is weaker at boundaries. Okay, therefore you have extra activity at boundaries causing boundary detection. Now region growing, and surprise, surprise, here you need to do the opposite. You need to put excitatory connections between neurons tuned to the same orientation. So how does this work? So suppose you're tuned to the left orientation and maybe there was some reason that you think you're important. Maybe there was a cue at that location. Then you want to tell your neighbors that they should also enhance their response because they're then also likely to belong to the same behaviorally relevant surface. Okay. So that's of course a bit problematic if you want to make a neural network because you want to put iso-orientation inhibition for boundary detection and iso-orientation excitation for region growing. So these effects will cancel. But there is a solution. And that is, we proposed many years ago that boundary detection might be closely associated with feed-forward processing, and region growing might only happen later during this feedback phase. And this shows you a very simple neural network model. So this is the input. Here are V1 neurons tuned to the left orientation, here tuned to the right orientation. So they respond to the background, they respond to the foreground. In reality, of course, they would be intermingled in a single retinotopic map. If you now sum these two activity patterns, you get a nice ring of excitation, boundary detection, right? Because of this iso-orientation inhibition scheme. Now the trick is to not only do this in V1, but also in highway areas with larger receptive fields. Okay, and if you then ascend the hierarchy, you'll find a level where the size of the receptive fields matches the size of the figure. And then you have neurons that are tuned to the figure orientation here left, and the neighbors are all kind of tuned to left orientation. They're not fed by feed-forward input, right? Because the neighboring orientation is right. So there is basically no inhibition. Psychologists call this pop-out. And pop-out is predicted to happen later at a higher stage if the figure is larger, okay? So that's all feed-forward processing. And now we're going to put in the feedback connections that are iso-orientation excitation. And under some conditions, you can then nicely take care of the fact that you want this feedback pathway, the spread of excitation, to be precisely confined to the figure or region, so that the enhanced activity is nicely confined to where the figure is. And this is quite consistent with psychological literature, who suggested that figure detection or pop-outs and boundary detection are pre-attentive. And the prediction of this model is that then the region filling should be an attentive process. It should depend on visual attention because it depends on the loop through the higher visual areas and then back to early visual cortex. Okay, so we tested this. So we trained monkeys on a task in which they saw a complex stimulus. They saw here a texture-defined figure, but they also saw two curves. Now, on some of the days, we call them texture segregation days, they were supposed to make an eye movement to the center of the figure. And on other days, they can completely ignore the figure and make an eye movement to the end of the curve that was connected to the fixation point, to this red dot. Okay, so this was done many years ago by Jasper Poort. And basically, on figure detection days, the animals were making an eye movement to the center of the figure, and on curve tracing days, they could ignore the figure. And if this is a receptive field of neurons in V1, we varied the location of the figure across trials. So sometimes the receptive field would fall in the center of the figure, but it could also be on the edge or in the background. And this is a replication basically of what Victor found many years ago. So you get a feed-forward response, and then if the receptive field's on the figure, you get more activity than if the receptive field's on the background. So what we're interested in here are these excess spikes, these extra spikes if the receptive field falls on the figure. Where are they and when? And to find out, we basically made this plot. So what you're seeing here are these extra spikes. So basically we're subtracting the background response. This is the spatial dimension. So here at 0, the receptive field is exactly in the center of the figure. At 2, it's on one edge. At 4, it's in the background. And here on -2, it's on the other edge. And then -4, it's on the other part of the background. Okay, so what you see here is the extra spikes. And you see that the first extra spikes happen at the boundary between figure and background. And then later is a gradual filling in of the center of the figure with enhanced activity. And this is when the animals were interested in the figure. So this was basically on the figure detection days. Now I would like to invite you to make a mental picture of your prediction of what this activity profile would look like if the animal is attending somewhere else. Okay, here's what you get. So you still get the boundaries because that's related to feed-forward processing, but you see that the region filling process that depends on the loop to the higher visual areas and back is much less complete. Okay. Now we also recorded from area V4, which is a higher level area with a receptive field size that is about matching the size of the figure. You see on figure detection days, activity or this difference response is higher than on curve tracing days. And here in the center of the figure, the response enhancement is already there early on because receptive fields are larger there. And actually we have a study in which we briefly, with microstimulation, silence V4. And what you then see is actually in V1, this response modulation also collapses, suggesting that some of this region filling in V1 here is dependent on feedback from V4. Okay, so let me summarize this for you. So you present a figure ground stimulus. Neurons are first coding for the orientations. Then just a little bit later, you get enhanced activity at boundaries, independent of whether you're interested in the figure or not. And then a little bit later, you get extra activity if the receptive field is in the center of the figure, but mainly so if you're interested in the figure and this figural, this region filling is much less complete if you're directing your attention somewhere else, as if these non-attended figures are left in a primordial state. Okay, so basically what we see is that early boundary detection is a pre-attentive process and this region filling depends on attention. Okay, now a very big question that we always had in the lab is we see this response modulations in V1, but people sometimes tell us, but they're very subtle. Are you really sure that they matter for perception, or are they just some sort of epiphenomenon? Okay, so to find it out, you would really like to silence V1. And it would be really nice to be able to silence V1 at a specific time point. So to let through the feed-forward response to higher areas and then shut it down. And we tried to use optogenetics in monkeys, but we found it to be very difficult. So basically, this is figure ground modulation in monkeys. So now we had to switch species. So we will go to mice, But first I want to show you also a result in humans. So we work together with epileptologists who see epileptic patients, and we use sometimes these electrodes through which we can also record single units. And we were able to record, I think this is actually multi-unit. So multi-unit spiking activity in area V3 of humans in the figure ground task. And you see that also in humans, figures elicit more activity in area V3 than backgrounds. Okay, so that's a replication of what we always have seen in the monkeys. Okay, now we go to mice. And basically what we find is that mice basically have the same effect. So figures elicit more activity in V1 of mice than backgrounds, even if you keep the image elements in the receptive field identical. Okay, so now we're going to go to mice for a while. And the nice thing about mice is that optogenetics works very well in mice. Okay, so this is what we train the mice on. So they see a stimulus, figure stimulus or a contrast stimulus on the left or on the right. If they see it on the left, they make a lick response to the left. And if they see it on the right, they make a lick response to the right. They can do this. And they're not as good as monkeys. So contrast, so this doesn't require any figure ground segregation. They're about 90% correct, but you see that for orientation, phase, and texture defined figure ground stimuli, they're only about 70% correct, but still much better than chance level. So we can start recording and see whether we get the same effects as we do in monkeys, which is the case. So here are contrast stimulus, so something in the receptive field versus nothing in the receptive field, not so exciting. Here's orientation defined figure ground and phase defined and texture defined figure ground. And basically we always find that figures elicit more activity in spiking than backgrounds. And you can look at the latency of the effect by subtracting the two responses. You see that the contrast response is basically feed-forward response after about 60 milliseconds. Orientation defined figure ground after 95, and phase and texture defined figure ground happen a little bit later. Okay, so that's nice, but now it gets really exciting. So we have optogenetic silencers in mice, and they really work very well. So this is work that has been done by Lisa Kirchberger. And so here's the idea. So this is the response by figure and ground, and basically we can completely knock out all the spikes from area V1 with DT-ACR2, which is one of these optogenetic silencers. Or we can postpone shining the light a little bit. So we let through the feed-forward response, the higher areas are informed about the stimulus, and only the late phase of V1 activity is abolished. Or we let through even a little bit more and more. Okay, so that's the logic of the experiment. Okay, we start with the contrast task, and this is maybe a bit of a complex panel. So what you see here is the laser onset, and here is when we don't shine the lights. So the animal only needs to detect contrast stimulus, no figure ground. And these gray symbols are different mice, and the green is the average across animals. So on average, they're about 85% correct. Now, if we start shining the light after 20 milliseconds or so, all the activity that is elicited by the stimulus is knocked out. Okay, no spike remains. And you see that activity drops, or accuracy drops. It drops to about 60%. So there's a little bit of above chance performance, even if you silence V1 entirely. And we speculate that this might be through the superior colliculus or some subcortical routes. Now then, if you start postponing the lights and we only shine the light after, say, 80 milliseconds, you see that the animals are basically back to ceiling performance. Okay, so just letting through the feed-forward response is sufficient for contrast detection. No need for this late response. In figure ground, it's very different. So if we shine light early on, knocking out all the V1 spikes, you see that the animals are now at chance level. And we have to postpone shining the light to, I guess, 120 milliseconds before they can do figure grounds. Okay, so apparently they do need a little bit of this late phase of their activity to be able to perform this task. So this is the first evidence that this late phase activity is absolutely essential for figure ground segregation. And we get a similar effect for the phase-defined figures, although now it's even later. Maybe it's 160 milliseconds or so before they can do the task. Okay, so we're excited about this because we think it's the first time that we could demonstrate that this late phase of V1 activity is important for visual perception, specifically in those situations in which you need to do figure ground segregation. Now, a small tweak of the experiment allowed us to investigate whether this response modulation that we see in V1 depends on feedback connections. We now put the optogenetic silencer in higher visual areas, and we recorded from area V1. So this now shows you the expression. You may know that in the mouse, there is a ring of higher visual areas surrounding primary visual cortex. And we then do for instance, the orientation defined figure ground stimuli. So if you don't shine the light in the higher visual areas, you get the normal figure ground response, more activity elicited by a figure. And if we then shine the light and we start shining the light before the stimulus appears, you see that in this example recording site, multi-unit recording site, the figure ground effect is much diminished. And if you look at the peak response, we find no effect on average, but if you look at the response modulation, you see that on average, if you shine the light, the figure ground modulation is much reduced compared to when you don't. Okay, so this is, I think, a very clear demonstration that part of this figure ground modulation that you see in V1 depends on feedback from higher visual areas. Okay, so I'll kind of summarize that we see in this task that the labeling of image elements with enhanced neural activity labels those image elements that need to be grouped in perception. And of course, this solves the binding problem. So no need for synchrony there. It's also interesting to compare what I told you before about this simple contrast detection task where the early visual areas just needed to propagate the information to higher visual areas where ignition occurred, as if the global neural workspace is confined maybe to parietal and frontal cortex. But in this more complicated figure ground tasks, actually V1 needs to be involved, actively participating in the perception, as if in those situations, maybe the global neural workspace even includes primary visual cortex when it's about figure ground. Now, the last 10 minutes or so, I would like to discuss how we are applying our insights into early visual cortex in the creation of a visual prosthesis for blind people, which is of potential importance for about 40 million people who are blind, although not all of them will benefit from a cortical prosthesis. I think a substantial fraction of them might. And we are targeting primary visual cortex. So we know that if you stimulate in primary visual cortex, which in the human is actually very unfortunately folded in the calcarine sulcus, difficult to reach. But if you put an electrode here and you stimulate, then a person, can also be a person who has been blind for several years, will see a dot of light. And the dot of light will occur there where you stimulate in the map of space. So we want to position 1,000 electrodes or more. So you can then create 1,000 of these dots of lights. We call them also phosphenes. They will appear at different locations in the visual field because you put your electrodes at different positions in the map. And of course, the trick is then to switch on the right electrodes and make patterns, like you can make patterns on a matrix board along the highway and make the subject see something, although it's going to be rudimentary compared to normal vision. So this is the system that we and others would like to build. I'm not going to claim that we're the first to do this because that's not the case. We have a camera, then a pocket processor that takes in camera images and translates it into brain stimulation patterns. We would like to send this wirelessly to a brain chip that then activates the right electrodes. And so that's what we set out to do. So we use these Utah electrode arrays. And this is the work of Xing Chen in the lab. Here is a connector, so we're not yet wireless. So this is something that we implant on the skull of a monkey. And you see there are 16 of these Utah electrode arrays. Each one is kind of connected to this pedestal with a bundle of wires. And we implanted this into monkeys. This shows you one of the monkeys. So here are the positions of the electrodes. And then we map out the receptive fields of those neurons. And so you see that, for instance, the cyan array has receptive fields down here. Here we're close to the fovea. So this, you see the predicted retinotopy here. Also in a second monkey where we replicated the effects. And it's nice to see the mapping experiments. So this is kind of simulated because, of course, during the experiment, you cannot really look at the arrays because they're under the skin. So here we project the activity on the arrays listed by a moving bar of light. And you see that the moving bar of light, of course, will go through the receptive field successively and you get a nice wave of activity. It's also remarkable to see this ongoing fluctuation, this spontaneous fluctuation of activity when there's no stimulus, right? And that does not reach perception. I think that's also kind of meaningful. Okay, you can also show the same results, but now in visual space. So we now map the activity at the location of the receptive fields on the screen. So here you'll see the bar, and you see that it's trailed by a wave of activity. And the time between the bar and the wave is, of course, the delay between the retina and the cell in primary visual cortex, which is about, well, to the peak of the response is 80 or 100 milliseconds. Okay, now for visual prosthesis, it doesn't make a lot of sense to be in recording mode. These animals were not blind, but the future prosthesis user will be blind. So you would like them to recognize electrical stimulation. So we trained the animals on a task in which we present the dot and then make an eye movement there, as I showed before. But now on some of the trials, we don't show a dot, but we show actually, we stimulate one of the electrodes, the primary visual cortex, hoping that the animal will make an eye movement to the location of the receptive field. And this is what happens. So every circle, small green circle here is center of one of the receptive fields, and the one that will flash in white is the receptive field of the electrode that we're stimulating. And in cyan is, no, sorry, in magenta is where the animal is looking. You see, he starts a trial by looking at the center of the screen, and then we stimulate one of the electrodes. And he goes approximately to the receptive field. You see there's a bit of a deviation, but that's actually our eye camera system. It is not completely well calibrated here. Okay, so you get the impression of how well this works. Of course, the big question, and that had not been addressed in previous work, is if you now stimulate multiple electrodes at the same time, can we write in pattern information into the brain? To find that out, we had trained the animals on a very simple task in which we present a letter from the alphabet. And then there was a choice menu, and the animal's task was to report a letter that he just saw by making an eye movement to the corresponding letter from the menu. Okay, and they could do this very well. We had trained them on this for many months. And then, of course, the exciting experiment is not to show this letter anymore, but instead stimulate a set of electrodes in the shape of a letter. And then we hope that the animal would make an eye movement to the corresponding letter in the choice menu. And this shows you one of the experiments. So we kind of imprinting the letter L by stimulating a number of electrodes, letter O. And you see that the animal is indeed doing the right thing. So when we saw this result, we were really excited because this worked better than we had expected. Actually, from the first trial onward, the animal is above chance level. So he's really generalizing from his experience in normal vision to prosthetic vision. Okay, so we are not yet there because, for instance, we are not yet wireless. It's interesting to know that together with Eduardo Fernandez, who has a lab in Spain, he implanted a Utah electrode array in the visual cortex of this subject. And she had been blind for several years. Also, he is not yet wireless. So there's this kind of known seroport from BlackRock, an array that is in her visual cortex. This is approximately where the array is. It's basically on the boundary between V1 and V2. And they then stimulated a set of electrodes here in the shape of a larger square, and she reported seeing a larger letter O. Here she reported seeing a smaller letter O, so that looks all really nice. Here she reported the letter I, which I already found a bit more surprising. And here she reported the letter L. So it's not completely straightforward. And I think what's going on here is that this is an array that is only 4 by 4 millimeters. So if you stimulate electrodes that are so nearby, you might get some unexpected interference patterns. So I think that that problem will be less if your electrodes are farther apart. Okay, and just I would like to end with this impression of what phosphene vision might look like, and to also warn the potential users of the system that it's not going to be terrific, certainly not in the beginning. So you can, the nice thing, you can test this in virtual reality, right? So you can just have people that wear the system that has a camera. So here you see what is shown here. So there's a camera here, and then there's a virtual reality system, and then there's this. Oh, you can test many of these processing steps that go from the camera image into a simulated phosphene pattern. But still, it's going to be better than seeing nothing, for sure. Okay, so I'll summarize. So in the first part of my talk, I talked about perception of stimuli with small contrasts. We looked at variations in internal signal strength that are explained by variations in feed-forward processing, and I talked about the threshold for perception. It's the sort of a bifurcation point that either produces self-sustained activity or not, related to working memory. The second part, we looked at conscious perceptions of textures, and then we see that it's not so simple. Now maybe the recurrent processes have to reach back all the way to V1. That might also become part of the global neural workspace that is there for stimulus dependent. And then the last part of my talk, I kind of suggested that we can use pattern stimulation of primary visual cortex to give back some rudimentary form of pattern vision to blind people. Now, last but not least, these are the people who did the work. So on the contrast detection task was Bram, Bruno, and Dev, collaboration also with the lab of Stefano Panzeri and Stan de Hane. The figure ground task that was done, well, by quite a number of people. So the genetics work was carried out very exquisitely by Lisa Kirchberger. And the last part on prosthetic vision was primarily carried out by Xing Chen, but also in a collaboration with Eduardo Fernandez and these other people. Here are the funders. It could be a good time to ask questions.
Thank you very much, Pieter. You opened up avalanche of questions here. At least in my mind, I have a few for each of your portions, their segments, but I usually give priority to students or other faculty. So let's see if others have questions. We'll let them ask them. Otherwise, if I have time, I'll ask mine. But thank you again. It was very stimulating. So
yeah. 1 second, Moshe, I would like to ask a question.
Go for it.
Have you tried to look on multimodal stimulations and to see that maybe you can have a lower visual threshold because there is integration of maybe audio stimulation at the same time, and then you can lower the threshold in higher layers. Yes.
Yeah, so I think that's a very good question. So there is work on that, right? On multimodal integration. and it's known from the superior colliculus, I think some old work by Stein and his collaborators, that superior colliculus cells are also sensitive to the, to say a visual and an auditory stimulus that coincide. Maybe it's best if they also coincide in space. So if the sound and the light comes from the same location. So it is an interesting research direction. We haven't done that, but my suspicion would be, as you probably it's also your suspicion, that it would go a bit better and you would lower the thresholds a bit. Yeah.
Any more? Okay, so maybe I'll ask a couple, but if you have a question, anybody just raise it.
Gadi just raised his hand.
Okay.
Yes, hi. Great talk. I was wondering if you had any thoughts about any ways to manipulate the random activity that ramps up and causes false alarms.
Whether you could manipulate that.
But I don't know, like if you had, I guess you didn't talk about any mechanism creating the random activity, but like controlling different rates of false alarms would be very interesting.
Yeah, so actually in the paper we looked a bit at that. So there's a part of the story that I did not go into, and that is related. We wanted to use pre-trial markers of brain state. And by looking at power and different frequency bands of the EEG, by looking at the eye and the pupil, we were actually able to create predictors of what the probability of, for instance, a false alarm would be. And we therefore could, based on the pre-trial activities, sort trials in those that were likely to produce a false alarm. This was all cross-validated. And trials that were less likely to produce a false alarm. And the difference was intriguing. So if the spontaneous activity level in all these areas was higher, then there was a higher probability of a false alarm. If the spontaneous activity level was lower, there was a lower probability of false alarm. As if, you know, if the activity level is higher, they're basically closer to the threshold for ignition, thereby increasing the probability.
Great, thank you.
Pieter, I remember from long time ago, a result from Mary Peterson, you probably know her, that stuck with me since I was a student. That I think she showed pictures, black and white pictures. It wasn't Mooney figures, but she showed some black and white upside down, and she showed that segmentation can be performed correctly without recognition, which I think is stunning, that if it's true and if it held water since, that you can know what contour and what edge belongs to what object without knowing what the objects are. Does this fit your thinking in any way?
No, I think what Mary showed is that objects help in the segmentation. So what she did was she had things like people upside down, and she demonstrated that. So because before Mary, people thought there is like a serial process. First the contrast come in, then segmentation, and then object recognition. And she demonstrated that also object recognition can guide the segmentation process. And I think that makes perfect sense. So actually I haven't shown it, but we have also started to do some psychophysical work in humans. And if you imagine that you have a picture with two birds, right? So one bird here and one bird there, and they could even be slightly intertwined or half occluding. And then we ask subjects, we presented on one of these birds or on two birds, two dots. And the task of the subject was to say whether these two dots are on the same birds or whether they're on different birds. And then you can measure their reaction time. And what you find, maybe to some people surprising, that your reaction time depends on the distance between the two dots. So there's a serial process, and that's what we call incremental grouping. So it basically as if this bird gradually fills up with enhanced activity. And the speed of that spread of enhanced activity also depends on the width of the component. So if the bird has a very narrow leg, the activity propagates very slowly over that narrow piece. Now, if you put the birds upside down, then subjects are much slower. So as if indeed, so in accordance with what Mary Peterson predicted, knowing about, normally we see the birds, well, straight, right, with the legs in the ground. And so we have a lot of visual experience with objects, and apparently that helps us in the segmentation process. And that's completely in accordance with what Mary Peterson said.
I see, great. But now that you said you're switching to move with doing psychophysics with humans, about your first part, I'm thinking about your definition of a miss and your analysis. And I remember a long time ago, my PhD hundreds of years ago was about subliminal perception. I remember this issue coming up in the analysis and really improving what one can find. So I can miss one of these quick presentations of a faint circle because it's too faint and the contrast is too low, even though I'm staring at it, or I can miss it because I've attended some other portion, other segment of. So these are two types of different misses. I would imagine there will be some weak signal in the first case where you do stare at it, but it doesn't cross your threshold, whereas the other, you just stare elsewhere in your attention at least. So not really staring, but allocating your resources.
Yeah.
And with animals, usually, obviously, it's harder for you to distinguish between it because you can't follow it with a four alternative first choice or some guesses. But I would imagine if you do this with humans, you can analyze differently misses that are a result of not looking versus missing of not crossing a threshold and then still having some weak signal.
Yeah, yeah. So I fully agree with you. And that could be the difference between, so remember that we have some of these cases in which activity levels are fairly high in V1 and fairly high in V4, and still the animal misses. So maybe that is one of these lapses of attention cases. Mm-hmm. And we have the other ones in which just the activity in V1 or in V4 is very low. And that might be one of these cases where, well, maybe the retina was in the wrong state. So what you're describing in sort of words of human psychophysics, I think that might map onto what we see in the monkeys in these different conditions. But of course, we can't ask the monkey, why did you miss that? Were you sleeping? So that's a bit of a downside of working with monkeys.
Any more questions? So last one from me, Pieter, about the prosthesis part. First of all, I wonder if, I missed the beginning of this part. Maybe you said, maybe you referred or addressed it, but the question of how long have they been blind? So if this congenitally blind or blind for a few years, I mean, you're an expert in plasticity, so there's a good reason to suspect that the neurons that you're about to stimulate already assumed another function or another type of selectivity. And the follow-up question for this is, have you considered looking at imagery as a guide for stimulation? So let's say you have some kind of either MRI or some other type of imagery and ask people to think about a car, and then you go stimulate in these regions, assuming these are not congenitally blind. So these are the two last questions from me.
Yeah, so I didn't say that, but this prosthesis will probably only work for people who have seen during early life. So say the first 5 or 8 years, so that they completely develop their visual system.
Yeah.
Because I think there's good work demonstrating that if you are congenitally blind, these regions, for instance, Ehud Sohari had some nice work on it, but there's other work as well, that V1 and is doing surprising stuff. They first thought it was related to braille reading. Later, they also found, Ehud found that it was even doing working memory for word lists. It's really unexpected and really odd, you know, V1 is doing word lists. But so in those subjects, if you then put electrodes hoping to activate pattern vision, you might do word lists of braille. You don't know what you're doing basically, so it won't work. So if people have been seeing for 5 or 8 years or something like that, then the evidence suggests that the critical period is over and the whole system is just dormant, it's just waiting for the next input that doesn't come. So people who made interfaces with the visual cortex made that sometimes for people who have been blind for more than 10 years, and it just works. So when the critical period is over, you can just plug in, it's the impression.
Okay. Well, because I forgot to record from the beginning, I didn't get to ask you, is it okay that we're recording your talk and putting it online? Yeah?
Yeah,
okay.
That's fine. Yeah.
Thank you very much again, Pieter, for a very stimulating talk. We appreciate your time and wisdom and data.
Thanks for having me.
Next time in Israel.
Exactly. Okay.
Thank you.
Bye-bye.
Bye.